In this video, I'm going to talk about the absorption process. So if you haven't watched the digestion process that I actually explained in the first part of the video, kindly pause this video right now and go and watch it. So I'm going to talk about the absorption process and how the amino acid is being absorbed into the systemic circulation. First thing that you have to understand is that absorption is basically what the process by which the body taking, or let me say the body uptake the amino acid into the what into the systemic circulation, and then it's actually occurred in the intestine, that is in a large amount. Absorption process takes place in the small intestine, but one thing that happens is that in the stomach, little, or let me say little or no absorption actually what occur in the stomach, right? Then because we also have what the stomach what cells that can also absorb what amino acid, and at the same time in the duodenum, which is actually the first part of the small intestine, absorption also takes place in the duodenum, but it might be a lot. Let me say very very little, right? If you have amino acid, right? If this is amino acid, right? Amino acid, these are the enterocytes, and as you can see, we have the villi that can also help in the absorption of the amino acid directly into the systemic circulation. So this is the systemic circulation, as you can see. This is everything that we have here as the systemic circulation. But one thing I want you guys to understand is that in the jejunum, which is the a specific part of the small intestine, right? So we have different type of cells that you are seeing here, which I'm going to zoom them. Look at this. I'm zooming them into this structure that you are seeing here. We have one structure, the second structure, another structure here. Then we have the other ones that we have at this side. But one thing I want you guys to look at is that after zooming this particular cell, one of them, we have everything that you are seeing here. So this is going to be what our enterocytes. This is another enterocyte. Then this is going to be another enterocyte. But as you can see, we have this finger-like structure, and this singular structure is basically called, let me simply say, they are referred to as what? As macrovilli, right? I'm going to call them the microvilli, right? They are microscopic structure that can that actually helps in the absorption process. Now listen, another thing that we have here is that, okay, I said these are the enterocytes. Let's basically call them enterocytes. That is the cells of the intestine. Then we have the microvilli, right? Then the next thing I'm going to talk about is that after the complete digestion of protein, we all know that we are going to have different products. And let's say the product we have here, we have what? Amino acid, we have what? Dipeptides, and also we have what? Tripeptides. But in this case, this is what we have. Then let me just basically come here. Let's say in this case, I'm having a dipeptide here, and at the same time, I'm having what? A tripeptide and also what? A dipeptide here. Let's say O minus and also what? NH3, right? Don't forget that we have something NH3 plus. So this is a tripeptide and this is a dipeptide. Now the next thing I want you guys to know is that another thing that we have here, let's say we have what? Amino acid, like we have amino acid here, bunch of amino acid, right? Different amino acid. It can be glycine, it can be any of them actually, but we have amino acid. How are these amino acid, tripeptide and peptide, how did they enter the enterocytes and get passed down into the systemic circulation? That's basically everything that we want to talk about in this video. How the enterocyte pick up this amino acid, this tripeptide and also dipeptide, and pass them down to the systemic circulation for the cells or the systems to actually mix up with them, because amino acid was actually very very important to the body, maybe for the synthesis of protein, which later in turn acts as an enzyme, or synthesis of amino acid that are actually responsible for neurotransmitters. Then another one that is that they can also undergo different kind of reaction in the body. They can undergo the amination, transamination process, basically for the formation of protein. You understand? Or for the synthesis of glucose through gluconeogenesis, right? Now let's just go ahead and talk about how the body actually absorb them. Now I want you guys to understand something. The enterocytes of the intestine. Now we have some specialized transporter here. These transporter, they are basically protein molecules that helps to uptake, or let me say internalize our amino acid. You understand? They help they facilitate the transport of amino acid from the intestine. This is the lumen of the intestine, right? So this amino acid, we're going to have all the amino acid here, right? That's basically what we're going to have. We're going to have the amino acid here, right? This is basically the lumen of the intestine, right? So these are the amino acids. We are still going to have some little bit of dipeptide. We are going to have what? Tripeptide as well, right? Tripeptide, dipeptide, amino acid, dipeptide as well. We are going to have them here, right? Then what happens is that this particular amino acid actually want to enter these cells that you are seeing here. These are the enterocytes, right? Enterocytes that you are seeing here, right? So that's basically what we want to explain. But one thing I want you guys to understand is that there are special type of transporter in this particular fluid border. So if we zoom everything that is happening here, this structure, if we zoom it, that's what we are going to have. This is basically what we are having, right? This finger-like structure that you are seeing here, these are them. These are the, look at them. These are the finger-like structure, which is the microvilli. Everything you're saying, this is it. Then this cell, this basically what is happening. Then down these parts is the systemic circulation, that is the blood value. You understand? That's basically what we have, and this what you are seeing here is the larger side that you are seeing here. Now, basically, what I'm going to talk about is that we have a transporter here, we have another transporter here, we have another transporter here. So this transporter that you're seeing here on the bridge border, they actually have their own mechanism, how they perform, how they help to transport amino acid into the enterocytes, right? They have how they perform their function. So first, I want you guys to understand is that this particular transporter here, I'm going to call it, let me just basically call it sodium dependent amino acid transporter. I'm calling it sodium dependent. This particular transporter depends on what? On sodium ion concentration, right? Then at the same time, I'm going to call this particular one that you're seeing here, I'm going to call it hydrogen ion dependent amino acid transporter. So these are the two special type of transporter that I'm going to talk about: the sodium dependent transporter and also the hydrogen ion dependent transporter. Now, one thing that happens is that this sodium dependent transporter basically helps to transport amino acids, right? Like its own action is basically for amino acid. It's basically to transport amino acid from the lumen of the intestine into the enterocytes. But in the case of this hydrogen ion dependent amino acid transporter, it basically helps to transport dipeptide and also like its major action is on tripeptide and also dipeptide. It helps to absorb them, transport them into the systemic, into the enterocytes. Now, this is what I want you guys to understand. Now, what happened here is that in the case of sodium dependent, right? In the case of sodium dependent transporter protein, or let me say amino acid transporter, is a type of secondary active transport protein. You understand? Because it requires what? Number one, it requires what? ATP and concentration gradient. And once again, sodium dependent amino acid transporter require what? It requires ATP, and at the same time, it requires what? It requires concentration gradient. Now, one thing that happens is that the ATP that is required by this sodium dependent transporter is not like, it doesn't require the ATP directly. That is the reason why that it is classed as a secondary active transport. You understand? Like active transporter protein. So sodium dependent amino acid transporter protein is a type of secondary active transport protein. Why? Because it doesn't utilize ATP directly. Now look at the downward part. There's a special type of a carrier protein here, a pump here. This pump basically performs a very good function here. Like it plays a vital role in the transport, or let me say in the absorption of amino acid. Now what happens is that inside the enterocytes, this particular transport or pump, like we basically pick up 3 sodium ion from the enterocytes. Is going to pump it out into the into the blood value. It will pump out 3 sodium ion, and at the same time it's going to pump in 2 potassium ion. As you can see, it's pumping out 3 sodium ion and pumping in 2 potassium ion. In that case, it basically requires it requires ATP for its optimum function, to perform its function. That is, at the point that it is picking up 3 sodium ion from the enterocyte, pumping it out into the blood value, it will be utilizing ATP. So what's going to be the name? The name is basically sodium. Since it is sodium and potassium, we basically call it We basically call it sodium ion / potassium ion ATPase, because it makes of ATP, right, to create a concentration gradient that is actually required to transport amino acid. And I'm going to explain the reason why. So this is a type of primary active transport, because it basically uses it makes of the ATP directly. But in this case, by pumping out 3 sodium ion from the enterocyte into the blood value, then there is a concentration gradient that's created. Why? Because the sodium ion level here is going to be very, very low. 3 sodium ion is pumped out. Then 2 potassium ion is pumped in. That means that we are going to have low concentration of sodium ion in the cytosol of the enterocyte. Now, the next thing that basically happens is that since low concentration gradient has already been created, then there is need for the enterocyte to pick up sodium ion from the lumen of the intestine, right? And then at the same time, at the point that it is pumping in, let me just simply say we have sodium ion here, right? Then at the point that it is pumping in sodium ion, right? Pumping in sodium ion into the enterocyte. We are pumping in sodium ion to the enterocyte. That's so that amino acid will also follow. At that point, amino acid will also follow. We are going to have amino acid. So this is amino acid. This is sodium ion. So at that point, sodium ion is pumped into the enterocyte. As it passes through this kind of transport protein, amino acid also follows. So this is a special type of co-transport, right? Because it actually transports more than a single word. The transport actually transports more than a single molecule. It transports sodium inside, and then due to concentration gradient of sodium, we have low concentration of sodium inside the enterocyte. We have high concentration of sodium outside, right, in the lumen of the small intestine. We have high amount of sodium ion here, right? We have high amount of sodium ion here. Can you see it now? Then we have low amount of sodium ion here. So as sodium ion move down the concentration gradient, it carries amino acid along. So here is going to be amino acid. We are going to have amino acid, right? Amino acid. Now this particular transfer can also do a special type of what's it called? A special type of function as well. As sodium ion, let's basically say this is the sodium ion. As it moves inside the enterocyte, right? Amino acid can be transported. At the same time, we can have the transport of dipeptides and tripeptides at the same time. Let's say these are tripeptides. Let me just put this inside here. Right? You can have inside this, right? So we can also have tripeptide as well. Now, what happens is that the enterocyte, the cytosol of the enterocyte, this enterocyte, right? Then the cytosol of the enterocyte contains some special type of proteases enzyme. Like it contains a special type of peptidase enzyme, right? So here there are some peptidases that are actually present in the cytosol of the enterocyte, right? They will basically cliff off they will basically act and cut off the dipeptide and also dipeptide and also the what's it called, the tripeptide. They will cut it into free amino acid. We are going to have free amino acid. Like once the dipeptide and the tripeptide, once they enter the enterocyte, there is peptidase enzyme present in the cytosol of the enterocyte that will basically act on the dipeptide and also the tripeptide and convert them into free amino acid. Then the free amino acid can now migrate and move freely, enter the blood vessel. For instance, so this is the blood vessel, right? It can move freely and enters the blood vessel. The free amino acid. That's basically what we have. So amino acid is available in the blood vessel now. Then it can be transported into the systemic circulation and get transport to the liver. That's the first case. Now in the second case, whenever sodium ion is entering, then we have transport of amino acid along it. Then this amino acid can basically move out of the enterocyte. Right? Why? Because we are going to have high amount of amino acid in the enterocyte compared to the concentration of amino acid in the blood vessel. So amino acids are going to migrate freely. They are going to enter freely into the blood vessel. Right? They are going to pass through some carrier protein and they will get transported. They will be available in the blood vessel. Then they can get transported they can move to the systemic circulation, as you can see here. Right? So from there they will get transported to the liver. Right. Now the next thing that we have here is that we have hydrogen ion dependence. Now in the case of hydrogen ion dependence, and before I even move to the hydrogen ion dependence, the transfer of sodium ion and the tripeptide is actually very, very minimal. It is not something that is common. You understand? Compared to that of sodium ion dependent, like hydrogen ion dependent. Right. In the case of hydrogen ion dependent, the transfer of dipeptide and also tripeptide is somehow rapid. Right. That is, they easily move along with hydrogen ion dependent transporter. This is a transporter protein as well. Let me just put the name here. We're going to have transporter. Right? So this is another type of what's it called? Another type of transporter. And then example, we can even have it to be, let me say peptide 1. That's peptide transporter. You understand? So this transporter here is a peptide transporter. It's basically moving what? Moving dipeptide or what? Tripeptide. Right? It will transport tripeptide and also what? Dipeptide into the what? Enterocytes. Now what happens here is that the hydrogen ion, right? Hydrogen ion that is found what? The lumen of the intestine, they will move into what? They will pass through what? Through the transporter and pass through what? And get inside what? The enterocyte. They will move into the enterocyte. Then from there, what's going to happen is that this particular dipeptide and peptide that you're seeing here, as this hydrogen ion move down, they will also move along with it. Right? So in this case, we are going to have dipeptide here. We're going to have dipeptide. This is going to be dipeptide, and I said we're going to have what? Tripeptide. So this is basically a peptide transporter. This is an amino acid transporter. This basically transports amino acid. Why this basically transports a peptide? Whether it is tripeptide or what, or tripeptide, that's basically what is transport. Now from here, let's say we have tripeptide, right? 3 amino acid joins together, and at the same time we have what? We have dipeptide. What happens is that, don't forget, in the cytosol of the enterocytes, we have specialized enzymes here, which are peptides. They basically act on peptides, converting them to free amino acid. Now what happened here? We're going to have the peptide here, the peptides here is going to clip up the peptide into free amino acid. We're going to have basically 2 here, right? Now at the same time, peptides are going to act on this guy as well, cutting it into 2 at the same time, right? Once they cut it into 2, once they split the bond, we are going to have free amino acid at the same time, right? So this free amino acid will move out. You understand? They will move out into the systemic circulation. This is basically what they are saying here. They will get transported out of the enterocyte, moving out of this enterocyte. Then they will be available in the blood value. They will pass through the blood value, then enter the systemic circulation. Right? This is basically what they are saying here. So we are going to move into the systemic circulation. So that's another mechanism at which, or let me say another mechanism by which amino acid and peptide can be absorbed by the enterocyte, by the intestinal cells. Now another thing I need to explain is that mechanism that the body can be used to transport amino acid. Right? Now what happens is that, don't forget, on amino acid, they are classified into, we have essential and also non-essential, right? Now that's why we have some amino acid that are in the body, right? And also, let's say we have some amino acid that we must consume them, we must get them through food, through diet, right? Now what happens is that, let's represent this particular red marker as the non-essential amino acid, and then let's represent this with essential amino acid, right? Now another kind of transporter of mechanism that the body can use to transport amino acid from the enterocyte into the systemic circulation is by exchanging amino acids. You understand? That is, one amount of this guy here, one of this guy here will be transported out, right? Then at the same time, one of this guy will move inside. You are going to have it here, right? So in that case, the non-essential amino acid will be transported out so that the essential can move in. So that's another kind of mechanism, but it's not as fast. This kind of mechanism whereby the body will exchange non-essential amino acid for essential amino acid. That is actually seen whenever there is no presence, whenever there is or let me say there is insufficient of ATP. Whenever there is no amount of ATP, there won't be creation of sodium gradients, sodium gradients in the enterocytes, because this transporter actually needs ATP for the creation of sodium gradients, right? Because transporting of 3 sodium out of the enterocyte and transporting of 2 potassium ion into the enterocyte actually require ATP. Now, whenever there is insufficient ATP in the body, right, there is no level of ATP, and the body needs to produce to absorb. Let me say the body needs to produce amino acid to produce protein rather. Now, what happens is that the body can actually switch to this mechanism using non-essential amino acid, right? Exchanging it with essential amino acid. and that can actually what happen whenever there is insufficient ATP in the body. So once sodium ion gradient salt is created, then the sodium at the lumen side is what is present what in high concentration compared to the one that's inside the cytosol of the enterocyte, which is what we are saying here. So once the sodium is moving inside, there is strand of amino acid inside at the same time, right? So at that point, amino acid will enter and move in freely. Once it gets to the enterocyte, it can pass through to the through a specialized kind of a special type of carrier protein here or transfer protein here and enter to the vein or capillaries. So once it enters into the blood vessel, then it will get transferred to the systemic circulation. Then all the amino acids present in the systemic circulation can now move through the blood vessel and get distributed to the different kind of cells that actually need them to carry out their function. Right now, one thing that I want you guys to understand is that amino acid, once it's available in the systemic circulation, it is most of them are first transported to the liver. Right, for them, like they move to the liver through a special type of blood vessel, which is actually called the hepatic portal vessel. So once they move through to the liver, they get transferred to the liver. Once they arrive to the liver, different kind of process can actually occur, like gluconeogenesis, deamination, transamination, different kind of process that ultimately lead to the let me say to a product will actually occur. Right, liver can actually store them. At the same time, liver can actually distribute them to different kind of cells that actually need them for function. Right, so that's just about absorption of protein. And then if you enjoyed this video, don't forget to like, share, and subscribe. It's very, very important. And at the same time, make sure you subscribe. It's very, very important. At the same time, subscribe.